my name is Ugochijioke. I'm a food scientist and I work with the National Root Crop Research Institute, Umudike in Nigeria. I'm also the product champion for Fufu in RTBfoods. I'll be presenting the main highlights and achievements on Fufu in period 3 on behalf of all the members of the team. The presentation will follow the outline listed below. We'll be looking at highlights from articles published in the International Journal of Food Science and Technology. We'll also be relating the results we found from proof of concept studies taken to understand biophysical properties of cassava genotypes that influence the rating ability. We'll also be looking at results that we generated using high throughput method for predicting quality trace of fufu and intact root. We'll also be relating the equations that we developed to predict dry matter and starch yield on intact root, as well as dry matter and total titrable acidity of wet fufu mash using the quality spec trek. We'll also be relating the results of studies on heritability and correlation we obtained from elite cassava clones on fufu quality traits in different locations within the southeast and west of Nigeria. We'll also be relating the impact of COVID-19, and at the end, we'll bring you target perspectives for period 4. The institutes and scientists involved in activities on Fufu in period 3 are listed on the slide seen here. So, on the major highlights of the first article we published, titled Quality Attributes of Fufu in Southeast Nigeria, Guide for Cassava Breeders, we looked at preferred traits across the different unit operations involved during Fufu processing. We looked at preferred characteristics of fresh roots, and we found out that big roots, smooth skin, white or yellow colored roots, and freshly harvested roots are preferred traits for varieties that we use for processing fufu. We also found out that while processing, the following traits were of utmost importance to the processors. These include the trait easy to peel, the trait high retenability, thickness of the mash while it is being stirred during cooking, and ease drying ability. At the point of consumption, we found out that the smoothness of the dough, the ability to be easily swallowed, its non-sticky nature, and mild or no fufu aroma are traits of preference. On the whole, we were able to identify three major traits as main traits that influence acceptance of the product. These include the ability to easily peel, the freshness of the root, and its high retenability. These were associated with the color, yield, organoleptic property, and overall acceptance of the cooked product. We also were able to produce another paper in the same journal, this time titled Understanding Cassava Varietal Preferences Through Pairwise Ranking of Gari and Fufu Preferred by Local Farmer Processors. Here, the study was done by using 276 pairwise rankings of roots and fufu products. We were able to deduce from the study that some attributes were rated very high for the fresh root, and these include its ability to have high dry matter, to have high product yield, to have high root yield, large size, easy to peel, have bright white color, and soften when rated. On the final product, the trait white creamy color, heaviness or density of the dough, smoothness, which is indicated by no lumps, firmness of the dough when touched, its ability to mold, its ability to preserve for a long time on the shelf, and its ability to swell when being cooked, and the stretchability of the mash are important traits to be considered on the final and intermediate product. However, we found out that for storability, the texture must remain in place. Fufu is not expected to sack out or sweat out water under storage. Some clones were identified as being highly preferred by the processors. These are materials that are being bred with the next gen cassava breeding project. The names of the clones are also on the slides I'm presenting. We also realized that these varieties were linked with textural and color attributes. But from the study, we were able to capture that bad texture and color are important traits that should be considered, that should be eliminated from varieties that will be used for fufu processing. Browning is supposed to be a negative attribute and should be avoided when we talk about products such as fufu. Based on our findings, we identified the high retin ability of the cassava as an important trait to study. Hence, we undertook the proof of concept study to identify biophysical properties related to the retin ability of cassava variety. The study was conducted using 14 elite cassava clones that were harvested from the Next Gen Cassava Breeding Program and a preferred land race, water. The materials were at 12 months of age. For the study, we also put into consideration the factor of the size of the roots that were peeled. This was also part of the traits that were identified during the WP1 study as quality attributes that influence the product of the product. So during the study, we had different sizes between small, medium, and big. These were all in 2 kg. The samples were further processed using 3 liters of water in different fermenting buckets, and they were kept at room temperature for about 72 hours, where after which they were grated, re-steeped, and sieved. The mash were then allowed to sediment, and finally the water to produce our fermented fufu mash. Some biophysical properties were captured both with the wet lab method and using the quality hand spec TREG. Some of the parameters that were evaluated include the temperature, pH, dry matter, cyanide, lactic acid, fiber, amylose, and amylopectin. These were evaluated both on the fermented mash and on the intact root. The rate of softening was measured using the penetrometer. This captures the study, the pictures while the study was going on. The results from the study on the effect of size on retinability of cassava roots showed that size or weight had no significant effect on the retinability of cassava roots as the days of fermentation increased. However, the study found out that based on penetrometer data, which was recorded as firmness, retinability is genotype dependent, and we're able to cluster the cassava clones into 3 contrasting groups according to the rate of softening. From the biophysical property evaluation on retinability, out that the penetrometer, which is related to the rate of softening, was negatively correlated to the amylose content of the fresh roots and dry matter of the fresh roots, but positively correlated with the amylopectin content, fiber content, and cyanide content of the fresh roots. Hence, telling us that the parameters vary depending on the materials that are being evaluated. We also went on to do further analysis on this same attribute and in CIRAD. The study also was also conducted using different cassava varieties. From the results, we found out that the genotypes varied in relation with their retinability, and we noted marked changes in firmness within 24 hours after retting. The study also tried to compare the rate of softening with the amylose content of the materials that were evaluated. And from the study, we found out that we found comparable amylose content between cassava genotypes with different retin ability. We also try to relate the rate of softening with the phenol content of the roots. The results showed us differences between the genotypes in terms of total phenol content. This study is still in progress. We also try to relate the rate of softening with the phenol content of the roots. The results showed us differences between the genotypes in terms of total phenol content. This study is still in progress. Within the year, we're also able to calibrate for to calibrate equations to predict the dry matter content and starch yield of intact fruits during fufu processing, as well as develop prediction equations for dry matter and total transferable acidity of the wet fufu mash. The study showed us that we were able to get good coefficients of determinations for predicting the dry matter and starch yield of intact fruits. We got values of 0.52 and 0.54 for the dry matter and starch yield of the intact fruits, while for the wet dry matter and total transferable acidity of the wet mash, we got coefficients of determinations for the predictions to be 0.8 and 0.66. This indicates that these traits can be predicted effectively using the handheld mirrors. These traits can be improved within the next period. Heritability studies were also conducted in different locations in Nigeria on elite materials to verify some some attributes for fufu and roots. Within the southwest. We worked in 4 locations, and we used materials at the UYT stage. A total of 288 materials at genomically selected varieties were used from set A, and a total of 216 clones were used from set B of the UYT clones. From the study, we found out that we were able to get that fufu has high yield across these locations. The value for fufu yield obtained was 25.9%, while the least value for fufu yield was 11.3%. However, we saw that variation in pure loss, and the values obtained ranged from 11.2 to 32.4%. Heritability studies were also carried out for fufu yield and related studies. And this showed us that the traits dry matter, fufu yield, and peel loss had very good heritability across these locations, indicating that they have strong genetic controls. Within the Southeast region, we did some phenotypic study correlation for the variables fufu yield, crude fiber, starch, and dry matter content. We had very good correlation between fufu yield and starch yield, as well as between fufu yield and dry matter. The study also showed us good correlation between starch yield and dry matter content of the fresh fruit. We were also able to, the heritability study we did in the southeast Nigeria showed us that, gave us good indication of strong genetic control for starch yield and dry matter content of the fresh fruit. Estimates of biophysical properties from next-gen materials used to process fufu also showed us that there were significant differences among genotypes evaluated for the traits dry matter, amylopectin content, total titratable acidity, pH, swelling power, and solubility. These are functional properties that were determined on the fermented fufu mash. The study shows that these traits have good heritability. However, we intend to continue this study in period 4 to validate the results obtained. The correlation result of this biophysical property shows that solubility is positively influenced by amylopectin and negatively influenced by amylose content of the material. Swelling power from the study was found to be positively influenced by the total acidity of the mash and negatively controlled and related to the pH. An increase in TTA was found to also correlate positively with amylose content, while an increase in amylopectin was found to cause a decrease in amylose content. So, based on the impact of COVID-19, we were not able to carry out our sensory evaluation study on fufu quality traits using trained panelists. This will be carried out in period 4. Period 4 activities for the year will include sensory evaluation with trained panelists and instrumental textual analysis using cassava breeding population and land races identified in WP1 activity 4 and 5. We will also continue our proof of concept studies to identify biophysical properties of cassava varieties that are related this time to the sensory and textural properties of cooked fufu. We intend to develop the SOP to determine instrumental textural properties of cooked fufu. We also intend to develop calibration equations to predict biophysical properties of the roots and wet fufu mash. These will include amylose, crude fiber, starch, and sugar. We will continue with our G/E studies in different agroecology to characterize biophysical and food quality traits of fufu processed from next-gen cassava varieties at advanced yield trial stages and at the NCRP breeding stage. In CIRAD, we intend to continue biophysical characterization of the retting ability of fufu with a focus on phenolic compounds within the period 4. At this point, I want to appreciate everyone that has helped to make this presentation a possibility. Thank you all.